Okay, welcome all to the first session. First session is building cell atlases. So this is essentially human cell atlas, Asia. What are the cell types, and what are cell states, and what's their biology, and so on. And then, as Jay mentioned, and Paradram and Pampan mentioned in subsequent sessions, get to infectious disease and other technology and other topics. We've got a fantastic speaker lineup here. Okay. A fantastic speaker lineup here. Our anchor speaker is Jimmy Yee from UCSF, whose work I've already mentioned. DMARCSLET formed the basis for IDA and many other projects, really a landmark way of generating single-cell data. Jimmy's a geneticist and a technologist. He's pioneered many single-cell and spatial technologies as a company in technology. He's, I think, profiling one of the most exciting ancestrally diverse populations in San Francisco, which will be really, I think, a landmark study looking at, okay, I don't want to give away too much of his study. And he's co-leads the Genetic Diversity Network within Human Cell Atlas. Jimmy.
All right. Well, thank you so much, Han, for the introduction, and the organizers for the invitation to come to beautiful Bangkok to share some of the work in my lab to scale existing single-cell sequencing technology so that we can profile large population cohorts. Here are my disclosures. All right, so I think most of us can probably appreciate that we're truly in a time in science where we can now profile hundreds of thousands, sometimes millions of cells per experiment, and there's been an exponential growth over time in terms of the number of cells that we can profile per experiment. And most of this has been enabled by a number of technological advances over the years. And so I'll just briefly describe an approach that my lab developed back in 2018 called MUC-seq, that has now allowed us to sequence many more cells over large population cohorts. And this has been described already briefly by Seyhan, but the simple idea at the time was that when you do a single-cell RNA sequencing experiment, or any time that you're doing single-cell sequencing where you're capturing some natural genetic variation within cells, for example, a TAC-seq, that you're capturing enough genetic variation that if you had access to some reference genotypes, we can very accurately assign each single cell correctly to the donor of origin, and also identify cases where two cells from different individuals are simultaneously encapsulated. And so we published a method to do this, and it was called the MUC-SLET back in 2018. And the consequence of this is that it allowed us to really increase the per sample, but also the per cell throughput for single-cell sequencing, allowing us to sequence large population cohorts, and in some cases, give access to groups that otherwise can't afford single-cell sequencing. And statistically, approaches like this allows us to multiplex or randomize our experimental variables so that we can better do downstream statistical analysis. So here's a study that my lab published earlier this year to sort of deploy multiplex single-cell RNA sequencing to scale to try to study or characterize the circulating immune system in patients with systemic lupus erythematosus, which is an autoimmune disease that predominantly affect biological females, and also has different clinical presentations in individuals with different genetic ancestry. And so our cohort is 165 patients and 99 healthy controls. It is one of the first times anybody has really looked at differences in both the molecular profiles as well as the cell composition between patients of East Asian as well as European ancestry. And again, we know that individuals of East Asian ancestry seem to present more severe symptoms. In total, we sequenced 1.2 million cells, or about 5,000 PBMCs per donor. In addition to doing single-cell RNA sequencing, we also did a limited number of samples using site-seq, so profiling surface proteins as well. Here's what 1.2 million cells look like in UMAP space, so we can identify the expected cell types in circulation. One of the results that I'm really excited about and really illustrated at the time when we did the analysis that single-cell RNA sequencing can in fact be really quantitative is if we just take a step back and group the myeloid cell populations the blue and green colored cell types together and call them myeloid, and then the red and orange and pink cell types together and call them lymphocytes, we can estimate the abundance of myeloid cells and lymphoid cells in circulation. And this is actually a measure that most of us get when we go visit a hospital and get a lab done. It's called the complete blood count. And so we can correlate what we estimate as myeloid and lymphoid abundance with what's reported at the time in the electronic health record of these patients at UCSF and observe very high concordance, right? So that single RNA sequencing data is in fact really quantitative in computing these broad classifications of cell types, or the abundances of these broad classifications. And then we can do downstream analysis and rediscover lymphopenia, that lupus patients have a loss of lymphoid cells in circulation and with a concomitant increase in the percentage of monocytes, but the actual absolute abundance of monocytes is not changing. It's just a loss of lymphocytes. But what's really powerful with single RNA sequencing is that we can go back in and identify the specific cell types whose composition is changing. In this case, it's the CD4 T cells, and in fact, the naive CD4 T cells that are being preferentially lost in lupus patients. And if you compare patients of East Asian ancestry versus European ancestry, you can see that most of the changes in the abundance of various cell types is pretty consistent, except for these naive CD4s. In patients of East Asian ancestry, there's even more loss of CD4 T cells in circulation, and that this is not confounded by treatment. In fact, we had a handful of patients of both ancestries who were not treated with any immunologics, and they still had a loss of naive CD4s in circulation. And so clearly, we now have identified a changing cell composition in these lupus patients. Lupus has been studied for decades, and one of the other features of lupus is that patients are presenting an acute gene signature that normally gets turned on in response to viral infections, which is called a type 1 interferon response signature. And you can see that if we treated our data set as though we had done bulk RNA sequencing, lupus patients, highlighted in these red columns, about half of them are producing elevated levels of these type 1 interferon stimulated genes. And we can't tell the difference between these two different color gene sets if we're just doing bulk RNA sequencing. But with single RNA sequencing, we can actually identify that these are two very different gene sets. This first set of genes are being turned on in all immune cell types, and that second set of blue genes are turned on selectively only in the cell types of the myeloid origin. Again, this information would be completely lost if we had done bulk RNA sequencing. So we have changes in cell state and changes in composition, and you can in fact start correlating these features. On the x-axis here is the degree of activation of those type 1 interferon stimulated genes in myeloid cells in lupus patients, and on the y-axis is the abundance of naive CD4 T cells. And you can see that these are inversely correlated. So the more activated myeloid cells are in a patient, the fewer naive CD4s they have in circulation, and we think that this is in fact consistent with the pleiotropic effects of type 1 interferons. They will increase the expression of type 1 interferon-stimulated genes in myeloid cells, but will also sequester CD4 T cells in tissue by activating CD69 and S1PR1 so that they don't get out of tissue. And we think what we're seeing is the proxy, what's happening in tissue in the blood. And these results are also consistent, actually, with clinical trial data on antibodies against type 1 interferons as a treatment for lupus. One of the most predictable features of that treatment is that the lymphocyte levels for these patients come back to normal. So that's lupus, and we're deploying MUC-seq in a number of other applications, obviously in COVID during the pandemic. We're also studying another autoimmune disease related to lupus called Sjogren's, and we're profiling a number of cancer cohorts, patients who are being treated with immunotherapy. And in collaboration with the CZI, we're also starting to now engage in profiling a healthy control cohort. And this is a cohort that we just collected in San Francisco. It's ancestrally diverse. It's 400 donors total, 100 of each ancestry of East Asian, European, African American, and Latinx genetic background. In total, we've sequenced about 1.6 million cells. In addition to getting the RNA, we're also doing the multiome here, so getting RNA and TAXI together. And this is just an early projection of data that's coming off the sequencer as of a couple weeks ago. We're still figuring out how to analyze the data. I'm happy to tell you more about it, hopefully in a later session. But what I want to do for the rest of the time is sort of motivate the need for new computational methods as we continue to scale the number of cells that we can profile per experiment. And so I touched on MUC-seq, which is genetic multiplexing to increase the number of cells that we can profile per experiment by about an order of magnitude. But there are new methods that are emerging, particularly combinatorial indexing approaches that promises to get hundreds of thousands to millions of cells per experiment. The way I like to think about combinatorial indexing is that if you think about the left as a standard 10x run of droplet-based single-cell RNA sequencing, you in fact have a lot of wasted reagents, and that if you do sample multiplexing, you're putting more cells into these droplets, but sometimes you'll have doublets that you can't do anything about, and you have to get rid of that data. With combinatorial indexing, you can make every single droplet essentially productive and get an order of 250,000 cells out of a reaction. And so clearly this approach should allow us to continue to scale the number of cells that we can profile per experiment. So what happens when we start getting data like this? Well, the question is no longer about do we identify a new cell type, right? Some of the analysis I showed is that we want to know what are the changes in cell state that's induced by case control status, maybe by genetic variation, or if we combine it with these perturbational experiments, that might be induced by a synthetic genetic perturbation through sgRNAs. And one of the unique things that's coming out of single-cell RNA sequencing is that we want to assess changes in the entire distribution of genes, not just changes in mean expression, but we can also look at changes in the variability of gene expression or changes in the co-expression patterns between pairs of genes. And so here I'm illustrating these sort of three different statistical analyses we can do. We can look for changes in mean expression for gene 1, or changes in the spread of gene expression for gene 2 that doesn't actually have a changing mean, or the changing correlation between these two genes. But the problem with single-cell RNA sequencing is if this is the data on the left, what we get is somewhere in the middle, where we have a lot of sparsity and we don't really know what to do with this. And there's been a number of computational methods invented to try to deal with this. The problem, though, with all of these methods is that we're not really modeling what happens within a cell versus what's happening through the sampling process of single-cell RNA sequencing separately. We'll try to model that together. And what we've done is built a method that basically doesn't make any assumptions about how genes are distributed within a cell, but we make very strong assumptions, and I think the correct ones, about what actually is happening as a sampling process from single-cell RNA sequencing. The key for the method is called Memento, is that there's two components to this. The first component is that we have now provided a set of method of moments estimators that precisely estimates, given the sampling process of single-cell RNA sequencing, what is the mean, variance, and correlation between pairs of genes when we actually get single-cell RNA sequencing data back. And the second is that we figure out a very fast way to shuffle data from single-cell RNA sequencing data to get really well-distributed p-values so that you can actually do statistical analysis and get results that you believe in. So here are just some figures illustrating how Memento performs in terms of estimating parameters. I'll just focus on the bottom one here. We've compared Memento estimates with what's reported by RNA-FISH, which is the gold standard. And you can see that even without imputation, so borrowing information from neighboring cells or neighboring genes, Memento is producing estimates that are highly correlated with FISH, even beyond estimates using methods that rely on imputation. Memento is really fast in terms of getting p-values that are well distributed, and because it's got really great p-values, in fact, improves the power of multiple testing over competing methods. So here's one application. Cheyenne discussed this a little bit, and we can certainly do this in our data set since it's a large cohort, is we can look for genetic variants that determine gene expression, maybe in a cell type specific way. Now on the right, I'm just showing what happens if you perform that analysis using pseudo-bulk versus using Memento, which of course now uses all of that single cell information rather than just collapsing that single cell, collapsing transcriptomes from many single cells into a pseudo-bulk measurement. We can also evaluate the performance of Memento. So here we've identified genetic variants that are associated with gene expression in all of the major cell types in circulation. This is from the lupus data. And we can ask whether or not those genetic variants that are associated with gene expression in specific cell types are also enriched for cell type specific cis-regulatory elements here on the y-axis. The answer is yes with Memento. And also you can see that with pseudo-bulk, but the enrichment is not nearly as strong. So, but in addition to mean expression, we can also look for changes in variants. So here is a genetic variant that's associated with the spread of gene expression for HLA-C. So you can see that individuals who are homozygous for A have the widest spread for the expression of that gene, as well as changes in co-expression. So here's two genes, JUNB and LYZ, where if you are homozygous AA, you have the highest correlation, and if you're homozygous GG, you have the lowest correlation. And this type of information will be completely lost, of course, if you do pseudo-bulking of the data. So just to summarize, I think multiplex single RNA sequencing is here to stay, and it can be used on large population cohorts. It's an unprecedented opportunity to capture variation in both composition as well as cell state. And we described the application of this in lupus, and that data is all available in Cell by Gene. And also a call out for new methods that I think we're going to need to analyze these types of data. And so I certainly encourage all of you to check out Memento at this GitHub. And we're hoping to put this on bioRxiv in the next week or so. And I'll just leave my acknowledgments up and take any questions. Thanks.
Thanks very much, Jimmy. Questions? Irene?
So is there, there is a minimum sample size for QTLs, and I'm wondering if there is any differences in the number of samples you need to call variance QTLs and co-expression and the usual eQTLs. Is there differences in power to detect those three?
Yeah, there's definitely a difference in power. We haven't done enough large population scale studies to know how that behaves as sample size, but it's definitely reduced power to estimate variance and correlation for sure, given the same sample size.
Okay. A quick question from my side. Really fascinated, Jimmy, by your result with Joseph that the eQTL effect sizes are consistent. Incredible. Asian ancestry individuals in San Francisco, European ancestry individuals in Australia, so far apart profile in different labs, but you found consistent eQTL effect sizes. On the one hand, that's an amazing validation of the method for inferring effect sizes. On the other hand, kind of almost disappointing. One would hope to find some differences. What do you make of that?
I guess my prior would have been that there's no different, that there would be very few places in the genome where the genetic variant actually will have a different effect size on different populations. I think the difference is going to be allele frequency, and I think this is certainly also true with GWAS data. There's been so many GWAS papers published on consistency of effect sizes. I'm sure there will be some exceptions to this, but I think most variants will have the same effect.
There's one more question there.
Yeah. I'm Yoshi from Osaka University. Thank you for your fantastic talk. So I have a question about the genotype-based demultiplexing. So did you evaluate its efficiency of the demultiplexing in a population with extensively high ratedness? I mean, some kinds of consanguinity.
Yeah. We worried about that initially, but there's actually data from some of our collaborators that you can demultiplex F1s.
Yeah.
So we think that there's so much genetic variant capture that you can even demultiplex first degree relatives.
Yeah. Thank you.
Thank you.
All right, let's thank Jimmy once again. Our next speaker is John Lowe. John is a senior principal investigator at Institute for Molecular and Cell Biology, where he serves as the program coordinator for stem cell regenerative medicine aging research. His lab does research on mechanisms of cell fate changes and develops new tools for deriving stem cell engineering. He received the Singapore Youth Award, A-Star Investigatorship Research Award, inaugural MIT TR35 Asia Pacific Award, and he's on the executive committee of the Stem Cell Society of Singapore and an elected fellow of the World Technology Network. Welcome, John.
Hey, thanks, Shyam. Thanks for the very generous introductions. I also want to congratulate the organizer for this very wonderful comeback symposium. So my name is John Lowe. I'll be sharing with you some of our recent work on single-cell dissections of the human's primary and organoid tissues and how we correlate them together. So my training is in stem cells biology, so I'm interested in how the cell fate are directed and how stem cells become a differentiated cell types. So we are very interested to identify all the transcriptional and epigenetic switches that control all these cell fate determinations. And along the way, we want to derive high-quality stem cells or tissues for therapeutic applications. A couple of years back, we worked with these clinicians from Singapore, as well as her collaborator from Mount Sinai to collect these human ocular samples. So the work is primarily driven by Pradeep from my lab, along with Kiyu, who is an informatician, working with him very closely. So we collected 6 human ocular samples of different ages, 20 to 80 years old, 2 female, 6 male, 2 female, 4 male, with no known ocular disease. And we also collected porcine samples from our collaborators from NUHS and of course, zebrafish samples from IMCB. And then we subject them to 10x genomic single-cell analysis. We didn't get very good dissections of the zebrafish samples because in the end we end up with epithelial cells only. So still need to work on the dissections of these very small ocular samples. But there are data sets out there, so we actually use the existing published data sets for data integrations on the zebrafish part. So in total, we have about 16 broad cells categories. And again, I want to emphasize that without single-cell analytics, you will not be able to see how diverse each of these tissue types can be, if you just collect the bulk samples. So we did the usual annotations of the cells, based on markers that were reported in the literatures. So for example, RPE65, highly concentrated in the RPE cell clusters. Also an indications that the RPE cells seems to be more homogeneous as compared to the other cell types. Very high expressions of RPE65, very concentrated clusters. Along the way, we also identified several novel markers, which we sort of validated with FISH and IF experiments on the retina sections. So, like what the earlier speech, one of the earlier speech alluded to, during the COVID-19 worldwide lockdown, many of the efforts were directed to the COVID-19 efforts. So we already had the data sets back then. We couldn't do a lot of functional validation experiments, and that's why we were looking very hard at the data sets, and we were able to, deposit these papers or manuscripts on the bioarchive, looking at ACE2 expressions, along with its partners, TMR, TMPR SS2, which is a very important associated protease. So we found that, although it was known back then that COVID-19 virus could infect the corneal epithelial cells, but not a lot is known on the cell types that were infected. So we found these very unique populations of conjunctiva cells expressing very high levels of AC2, but not a very large portions, a very unique populations of it that also co-express these associated proteins. So we then came up with these computationally, the predicted pathways for the virus to infect these ocular system through the conjunctiva cells, suggesting that COVID-19 virus could infect the eye and make the eye the reservoir for the infections or its replications. We are a stem cell lab, so we, whatever tissues we should profile, we will always be looking for stem cell populations. So we made use of this pipeline known as SENT, stands for single cell entropy pipelines that allow you to look at the potency states of the cells. So we are particularly interested in the iris and the ciliary bodies because there have been quite a number of studies out there that suggest that some of these cells do have multi-potencies in the ciliary bodies. And so we ran this pipeline and we identify with very high stem cell potencies, these very unique populations known as the putative stem cells. We just call them that because we simply do not know what their function is at this point of time. And then the pigmented ciliary body cells also with very high stem cell potencies level. And number 4 would be these CoL9A1 high ciliary body cells. So we tried to do some experiments. We went back to the frozen samples, iris tissues, thaw them, try to follow through lineage analysis, their stem cell potency, whether they could go on to one lineage versus another. So the work is still undergoing. I hope to be able to present to you these unique stem cell populations in the next updates for the HCA Asia meeting. But switch gear a bit, we are always interested in how cell fates are regulated. And that's why we perform this Reglons analysis of the different cell types within the ocular systems. So, these annotations M1 to M13 are the different module systems. And if you look at this putative stem cell population, so indeed, M11's module, it seems to, have a very unique expressions of certain transcription factors as well as the regulatory of its putative downstreams targets. But let me turn your attentions to these RGC cells, also very, very specific in terms of the Reglons that governs it. So M1 is the Reglons that governs the RGC population. So if you look at some of the genes or transcription factors that define the RGC population, so some of the factors are known factors that are markers for RGC. We also identify quite a number of novel unique transcription factors like KRF7, EBF2, EBF3, etc. And if you turn your attentions to Muller-Glaire cells, for example, so very similar to Schwann's cells in terms of some of its, Reglons network, suggesting that because maybe Schwann cells were from the Glier origins, so that's probably one of the reasons why they share a lot of overlappings in terms of their Reglons analysis. So we then went on to build a network of these regular maps for the ocular systems for the first time, taking into account all the cell types within these Reglons tissues. We found that many of the factors that are involved are from the KRF family, from the Zig family like Zig2, Zig3, also from the EBF families. We wanted to sort of functionally validate some of the targets. So we picked KRF7, which is one of the Reglons identified for the RGC. So we did RNA phage, we did immunofluorescence experiments, and we show, indeed, KRF7 is expressed in the RGC cell layers. We then sort of established these differentiation systems from human embryonic stem cells to the RGC lineage. So Chen from my lab was doing the experiments along with Pradeep. So we were able to differentiate them up to day 32. You see a totally different morphology after undergone the RGC lineage differentiations. So we then sort of perturbed the systems. I must say that although we differentiated the cells to RGCs, the cells in the culture were very heterogeneous. Either we have not mastered mastered the protocol very well, but when we did single cell analysis, we found a mixture of different cell types. So when we look at this control vector knockdown single cell analysis of cells that were differentiated to the RGC lineage, we found that there were cells that are simply neuronal, there were cells that are 807 neuronal cells, but only maybe 25% of the cells were RGC in nature, so RGC precursor cells, RGC cells. But still, when we knocked down KRF7, we found that there is a downregulation or decrease in numbers of the RGC cells. Some, maybe down to 60% of the vector control. But when we overexpress, it seems that the effects is more drastic. So again, we have done single cell analysis. In overexpressions of the KRF7, you get, maybe about 3-4 of the overexpressions of the empty vectors increase in the KRF7. So interestingly, the precursor cell doesn't change that much, but it's really the mature RGC cells that change with the overexpressions of KRF7. So it seems like KRF7 is important for the maturations of the RGC cells in culture here. So at this point of time, Chen also developed these organoid systems, trying to use it to functionally validate some of the findings from the primary tissue experiments. So she managed to grow the organoids very patiently for 200 over days. She told me that the trick is really to be able to see laminated neuroretinal layers. So those would be the very good organoids that you could dissect and then perpetuate in culture. So we did some experiments with these organoid systems. So we collected them for single-cell RNA experiments. We built this lineage-specific transcription network. So we then try to identify what are the earlier cell types that sort of appear during the organoids differentiations, and which are the cell types that appears much later. So it seems that RGC cell types came up pretty early, so detectable by about day 52. So we then decide to sort of validate some of our earlier targets in the RGC M1 regulons. So we also found that, through this trajectory analysis and differential gene expressions analysis, comparing to the fetal RGCs, genes like KRF7 already described in our primary tissue single-cell analysis, EBF1, all these usual suspects turn up again to be very specific to the RGC lineage, even in the organoid systems. So she did a very quick and dirty experiments by using CRISPR of the EBF1, put it into the organoids. So the efficiency of the infections may not be very high, but she did detect downregulations of the EBF1 mRNA. So this is one of the markers for RGC, also went down. So sort of validate importance of EBF1 in the organoid systems. So in summary, we identify critical gene transcription factors like KRF7 and EBF1 to play a critical role in the retinal ganglion cell cell fate. So moving forward, she's trying to use these to actually perfect a methodology for single-cell CRISPR screening in the organoids. We're also developing a multi-omics spatial transcriptomics on human organoids and tissues. I'm looking forward to many collaborations from this meeting, and I just want to thank my collaborators and funding agencies. Thank you so much.
Thank you, John. Questions? Wayne?
Hey, John, that was a great talk. So I know this maybe wasn't the main focus of the results you wanted to present, but could you talk a little bit more about what insights came from doing both the porcine and the human cells, right, in terms of cell type differences, or what would you see? And what are you expecting to see with the zebrafish data when you finally get that point?
Thanks for the question, Swin. So I didn't present the conservation data which we presented, which we actually included in the manuscripts. So there were some regulons that are different between the humans and the porcines. So we did quite extensive studies on the divergence in terms of their regulons, and obviously zebrafish is even further away. So we were using these conservation studies to sort of inform us that certain regulons or certain transcription factors are really key, and that's why they were conserved. So KRF7, EBF1 are two of the critical transcription factors that were highly conserved between the different species, and that's why we focus on them.
Okay, great, thank you.
Thank you.
Thanks again. A very short question. And a short answer, please.
Yeah, it was really nice to see the Herculean effort by your student to get those organoids going. Can you quickly discuss like how closely does the organoid represent what you see in the human eye?
Yeah, so that's one challenge of trying to grow organoids from human embryonic stem cells because they are often embryonic and fetal in nature. So despite the fact that you manage to culture them for hundreds over days in culture, it just doesn't recapitulate the entire developmental process that took years and tens of years. So in fact, but they are very good as a model for early development. So we were pleasantly surprised that KRF7 and EBF1 were also present in the organoid systems because we were expecting that other fetal transcription factors may be even more important. Yeah, thank you.
Thank you very much once again, John.
For the next speaker, I am happy to introduce Dr. Jong-un Park. Dr. Jong-un Park is an assistant professor at Graduate School of Medical Science and Engineering at Korea Advanced Institute of Science and Technology. He has worked extensively on developing computational methods and databases for integrative single-cell data analysis, building single-cell-based diagnosis platform, and understanding the development and aging from the integrated cell atlas. Dr. Jong-un Park, we look forward to hearing your talk.
Thank you very much for a nice introduction, and I'm really honored to share my recent work with this wonderful community. And today I'm going to talk about the topic about systemic dissection of tumor ecosystem at single-cell resolution across 30 different cancer types. And so let me start by explaining what I mean by tissue ecosystem. And recently, there's a trending AI program which we just give them a word and then they give us back an art. And I asked this question: what do you think about cellular heterogeneity of the human tissue? And it gave me this pretty picture. I really liked it not only because it was pretty, but I think already AI is capturing the real essence of human tissue, which is that firstly, the AI knows that it is composed of single cell, which is what we are all thinking about. And then also another interesting thing is that it is also understanding that there's a pattern that is made by the cell-cell interaction of the specific cell type, and that is recurring across the tissue. So you can see the pink ones recurring and the orange ones recurring. So I would call this recurring cell-cell interaction program in human tissue as a tissue ecosystem. And then I think we are really at the right stage to think about these recurring tissue programs because Human Cell Atlas has been so successful due to all of the participants. And now a lot of data are being collected in the Human Cell Atlas DCP. So there's a huge data set, and now our main task is actually lies on the data integration task, how to harmonize all the data coming from different tissue, and how can we interpret that. And to start the procedure, I thought that cancer could be the good starting point because I really thought that cancer is actually a self-sustainable tissue ecosystem that sort of gets generated in all the different tissue in our body. So it comes from all the different organs, and based on their origin, they have a slight different sort of tendency, which we would call as an intertumoral heterogeneity. And within the tumor tissue, we can really see all the important ingredients of the human tissue, including malignant epithelial cells and fibroblasts, and also all the immune cells. So it is a really good sort of experimental point to think about what is the generality and diversity of our tissue environment. So we made this data collection, which is a single-cell data collection. So it's a meta-analysis. And there are many pan-cancer meta-analysis ongoing at the moment. And there has been a beautiful work published by, for example, Dr. Jamie Zhang's group on pan-cancer T cells and pan-cancer myeloid cells. But at the moment, because we are really interested at understanding tissue ecosystem, we really focused only on unsorted samples that has undergone no FACS or MAX sorting, so that we know that each data set should represent the tissue in the mostly unbiased manner. But we also understand that there is a bias according to the single-cell experiment by itself, but we are at the moment neglecting that. And then we collected only the data set from the single platform. This is not the advertisement, but from our demographic search, 10x Genomics was dominating one, so we chose that as a platform. And then we collected tumor tissue and also matching just normal tissue data and also healthy normal data. And that sort of comprised in total 4.4 million cells, which has been manually curated for their metadata. So they now have the same sort of nomenclature for all the metadata. And it contains about 100 cancer data set featuring 1,000 cancer patients and about 500 normal donors. And it encompasses like 30 different cancer types and maybe around 20 organ types. And after collecting this data, what we did was trying to project them onto the same space. So it took a lot of time, but this is our final UMAP structure, where we could also sort of roughly annotate them. Like these are epithelial cells, these are T cells and B cells and myeloid cells and all the other cell types. And you can see that some organs are well mixed on certain cell type, but epithelial cells, especially they show the organ specificity. And then for the tissue type, we have overrepresented tumor tissue, but also there are like normal tissue, and some are mixing to each other. But at this moment, we also realized that we are losing a lot of resolution by integrating data, and it's somewhat inevitable because this data set contains a lot of batch effect. It's coming from the multiple sort of institutes. So we thought about what would be the best way to sort of retrieve that lost resolution while we are integrating all the data into one. And then we came up with this approach, which is also now being widely adopted in the single-cell field, matrix factorization, especially non-negative matrix factorization method to decompose single-cell data. So we performed NMF across multiple samples per cell type, which generates a lot of NF factors. And then we project them onto the UMAP space to see what's happening there. It was quite really helpful because we can now see what kind of factors are there, which ones are sort of correlating with each other, and which ones are sort of organ-specific. And based on that, we also came up with the informatic pipeline to rule out some factors that are quite boring to us, or rule out some factors that are obviously being generated by the soup contamination. The usual, like the single cell data, I would say, usual problem in the single cell data analysis. And then we got the finalized NF UMAP structure that looks like this, and then we utilized that. So eventually, these annotated NF modules, we call them as cell states. And then we came up with about 70 cell states that can be found across different cell types. And then we also added some important cell states that has not been discovered in our study, but has been well described in other studies, which in total result in about 100 filters to understand tumor microenvironment in higher resolution. So let's zoom into the process that we did here. So these are the NMF UMAPs. So these are not the cells, these are gene sets that has been projected onto the UMAP space. And this is quite useful because now gene sets looks like cells, and we can just intuitively annotate them using the same approach that we used to annotate cells. And these are like the cell state annotation. And once they have been annotated, we sort of average the profiles and then call them as cell state signatures. And then obviously we can also find marker genes for these cell states. So these are like the marker genes that represents each of these cell state. And then here I adopted the method that is developed by Sham, and this morning Sham also mentioned about second version of RCA. So this referent component analysis allow us to project our cells onto this cell state space. So we utilize this cell state space that we have annotated, and then we project cells back onto that cell state space, which resulted in this cell UMAP. And by doing so, what we could do was linking the cell state with cell types. And obviously, there are some cell state that is reflected by just a single cell type, but then there are also the cell states that is represented by multiple cell types. So they might have been missed during if we just has focused on the cell type markers. So this is what we have done, and we annotate a lot of cell states as like this. And then after that, we started to ask the specific question. For example, what kind of cell states are truly niched in cancer? And for example, here we are sort of measuring the ratio of observed and expected ratio for the NF factors. And like this one, and if you compare these two, like highlighted one, these two are the cell state reflecting the inflammatory fibroblast. But the one with specified the marker of Wnt5a is highly tumor specific, as you can see here. but the other ones are also observed in normal tissue. So we can really distinguish what is the general tumor program, what is the tumor-specific program. And then we can also ask this same question about the organ specificity. So whether these cells, each cell state, are they specific to each organ or are they generally highly observed? And for example, for this desmoplastic fibrous signature, you can see the pediatric pancreatic cancer has a quite high cell state score for the desmoplastic fibrous, and also for this another cell state for the 6L10 high fibrous, you can see there's a different demography for different cancer types. And we can also ask whether these kind of cell states can be used as a filter to understand patient survivor or response to the therapy. And for that, we need a lot of population data. So we went back to the bulk RNA-seq data set, like a large TCGA cohort, which has survivor information. And then we deconvoluted the data using the cell state information that we have defined. And as you can see here, we can note many interesting sort of patterns. For example, for the low-grade glioma, any cell state related to the fibrous, if they show up in high in patient, that predicts the shorter survivor. Whereas for the renal cell carcinoma, if we see any endocellular signature coming up, then that sort of predicts the longer survivor for the patients. And again, the tumor-specific inflammatory fibrous signature here, it predicts the shorter survivor for certain cancers, but not all of them. And we are at the moment not sure whether this is due to the still lack of the statistical sensitivity or not, but it also shows us that there are differences between tumor types about about each factor, whether they're contributing to the survivor or not. And of course, we did this for all the cell type specific cell state factors. For example, for epithelial cell states, we have defined these cell states. And we also added some known features like T-cell exclusion program and hypoxia signature or PDL1 and PD1 checkpoint pathway, which you can also see that which tumors are really expressing high level of these kind of features. And for example, in renal cell carcinoma, you can see really, really high signature for hypoxia, which is well known because they sort of have a genetic mutation to sort of upregulate HIP to alpha. And then we can also do the survivor analysis where we learned that actually, as you expected, the differentiation of cancer usually predicts the shorter survivor. And if you retain the tissue-specific markers, then it predicts the better survivor in general. And if you zoom into each of the cases, then you start to observe many interesting new findings. But for the sake of time, let me jump onto the main dish of today, which is investigating the co-occurrence of these cell states. So this is going back to our original question, understanding tissue microenvironment and tissue ecosystem. And what we are doing here is we are just checking whether certain T and NK cell states are co-occurring with any of the myeloid cell state. And as a sanity check, what you can see here is the interferon response signature in T cells and myeloid cells, they co-occur, which makes sense because if the tissue is being receiving a lot of interferon signature, then T cells and myeloid cells should be both responding to them. But what is interesting is that on the left side, we have a normal co-occurrence map of the normal tissue, and on the right side, we have a co-occurrence map of the tumor tissue. And as you can see here, this interference signature is sort of correlated and co-occurring with more diverse T-cell signatures, which actually includes the exhausted T-cell signature and regulatory T-cell signature. So this shows that the response to this interferon response differs between normal and tumor tissue ecosystem. And we can summarize all these findings into this kind of cell state interaction map, which cell states are co-occurring with each other. And what was interesting to note was that we start to observe some kind of modules here. And this is tumor-specific network. And you can see many well-known factors for the tertiary lymphoid structure are sort of co-occurring within this module. And there are some novel factors that is now included in this module. So we questioned, because recently many papers have suggested that this tertiary lymphoid structure, especially the germinal center B cell signature, can contribute to the cancer immunotherapy. So we checked whether these components are predicting the response to the cancer immunotherapy. And indeed, all the module members showing the co-occurrence in this data has a like predictive power about the responsive for the cancer immunotherapy, not only in our cohort for the lung cancer, but when we observed throughout multiple pan-cancer data set, we consistently observed this pattern. And then finally, we asked the question whether then this kind of like recurring tumor ecosystem, can we use that to classify the tumors regardless of their organ of origin? So we sort of projected all the cancer patients' single-cell data onto the UMAP space using these features as a main feature. And then what we could see is this ecosystem tree, what we named as ecosystem tree, is knit with all the TLS components. And it contains multiple cancer types, but actually head and neck cancer is specifically niched with this ecosystem tree. So we kind of thinking that depending on the organ of origin, this kind of there might be skewing in which cancer falling into which system like better. So in summary, we constructed the pan-cancer single-cell atlas, and by manually annotating these NMF modules, we generated 109 features to understand tumor ecosystem, which gave us the tumor co-occurrence map, or which cell states are co-occurring, and that also led us to identify finding all the components of cancer TLS, and which are all predictive of immune checkpoint response. So that's it, and I would like to thank our lab members and also our collaborator. So this work has been mainly done by Junho Kang and Junyoung Lee, two really talented graduate students from our lab and Dr. Jung-Kyun Ch's lab. And our immune checkpoint therapy cohort has been received by Professor Seyhan Lee's lab in Samsung Medical Center. Thank you very much.
Thank you very much for the exciting talk. So we can take probably one quick question from the floor. Yes, please.
So thank you for exciting your work. So I'm very curious about your approaches, the NF-based approaches, because as you know, the NF is basically the summation of the non-negative basis vectors. So that means the library sequence, the depth, and also the complexity will be the very strong confound factor when you deconvolute the large-scale, the single-cell matrix into the multiple bases. So how could you ensure that these are not the main factor to define such a different models through your analysis?
Thank you very much for the question. So there's two answers. First, we run NMF per samples. So most of the biases across sample is ruled out. And also, when we are sort of summarizing this, we ruled out some of the factors that is coming from those kind of factors, like sequencing depth and things. So there's an additional filter that was applied on the later step.
Okay, thank you.
So thank you very much. The next speaker is Dr. Christine Wells. Dr. Christine Wells is a professor of stem cell systems and director of the University of Melbourne Centre for Stem Cell Systems. She is a genome biologist with a focus on the differentiation and activation of mammalian cells, and a particular interest in the intersection between innate immunity and stem cell biology in tissue injury and repair. Please, Christine, the stage is yours.
Thanks very much for the introduction, and particular thank you to Ponpan, Varadum, Anaya, and Vim, and the local organising committee for putting on just a fantastic meeting. I really enjoyed the day, and I know the rest of the meeting will be great too. So I want to talk to you a little bit about computational and stem cell models of a very particular type of immune cell, the myeloid cell, and two classes of myeloid cells, the dendritic cell and the macrophage, which are resonant in every tissue of your body and have very distinct roles in each of those tissues, everything from helping regulate your body temperature in brown fat through to keeping your mind clear and active through synaptic pruning of the neural synapses, all the way through, of course, to tissue homeostasis repair, detoxification, and response to infection. So a big task. And yet when we've seen the immune cells, the myeloid component of the immune cells mapped from our blood, what we see is this amorphous mass. We rarely see any of these specialist roles, in part because we're only sampling the blood, and in part because the resolution of information that we're getting may not be captured properly from the sparsity of the single-cell data that we're getting. So my lab is approaching this problem a few ways. The first is we've tried to build up some reference maps from old-fashioned techniques, really counting on the fact that these very redundant platforms now, bulk RNA sequencing and so forth, are often accompanied by very rich experimental metadata, and we can draw on that rich experimental metadata to inform the single-cell studies and gain better annotations of the cells that we're interested in. And also we are testing how closely we're able to mimic these tissue functions by developing induced pluripotent stem cell models of myeloid biology in a dish. So these are iPS-derived macrophages. These were made in the lab by the wonderful Nadia Rajab. And you can see that they're doing all the things that a macrophage should do. They are expressing the cell surface markers, which are typical of this blood cohort, but they're housekeepers, right? You can see them wandering around the dish and cleaning up other cells that are dead or dying. They're really creating a homeostatic environment there. But still, they've been derived completely artificially in a dish. They haven't gone through a developmental program in the bone marrow or the fetus. And so we have this question, how similar are they actually to the real thing? This is just very quickly to tell you what our computational pipeline is to bring together data from different experimental platforms, including between RNA-seq and single-cell data. Jani Choi is the team leader of this work, and he will be at the meeting as soon as his delayed flights get him here, but I would encourage you to go and talk to him about this aspect in more detail. So when we look at our dataset projections, we can put single-cell data generated by Nadia and look at where they sit across the atlas. You can see that we've got very rich annotations according to tissue type, according to how they've been derived. So an in vivo sample has been sampled directly from the tissue, an ex vivo sample has been cultured for some time, and an in vitro sample has been generated entirely in the dish. We can look at gene expression across this and see how it behaves across the atlas. We can look at the number of cells or samples that have contributed to those patterns so we can get very reliable reproducibility. This is a cross-experimental series, not across individual cells at this point. And when we look at where the data from Nadia's macrophages sit, you can see that they are sitting very much in that macrophage cloud here. Sorry. That macrophage cloud here. Oh, it's a very fancy laser. And you can see that they are really very similar to other in vitro derived cells, but also coming very close to macrophages from tissues such as the synovium and blood. So we've used this ability to project single-cell data onto our bulk atlas to also annotate myeloid subsets in public single-cell data, including 2 studies that were published some years ago now, again from blood. And what you can see very clearly are that not only are we able to identify macrophages and dendritic cells, but we're getting very clear stratification between subsets of these cells, such as the conventional dendritic cell type 1 and type 2, and the plasmacytoid DCs, which are often quite rare in single-cell data sets and actually can be hard to identify based on just a small number of marker genes. I want to give you an example of why this is important to do, and the example I want to give you is, again, a study that's been published by another group who have tried to reprogram fibroblasts to get to the conventional DC1 subtype. This is a very rare cell type in your body, but absolutely critical for anti-cancer surveillance because it cross-presents the tumour antigen to the CD8 T cells to elicit a proper immune response. And there is some speculation that cancers arrive when our DC1 subsets become exhausted and can no longer perform that surveillance mechanism. So this group very cleverly tried to derive the DC1 subset from human fibroblasts through direct reprogramming. You can see that they've added these transcription factors to the fibroblasts to get to their DC1s, and they're phenotyping them both on FACS expression of the HLA-DR marker, which is part of the antigen presentation machinery, and on comparison of their reprogrammed cells to primary blood DCs. Here you can see the DC1, DC2 and PDCs they've isolated from blood. And you know, if you just take this TSNE and correlation score at face value, it looks like they got to where they wanted to go. But if we project this data onto our myeloid atlas, then what you can see is that they haven't quite reached there. So I'm just showing you how simple it is to project your data onto this atlas. You just need two types of files, one which is providing the expression matrix, and the other which is providing the sample information, and then this gets uploaded onto the atlas reasonably quickly. And we can see not only where does it project in relation to the myeloid classes, but also is it more similar to any of the myeloid tissues. And what you can see pretty clearly are here at the bottom are the primary cell types that they had included, and they are sitting down with our conventional dendritic cell clusters, but their reprogrammed cells really are not getting anywhere near that DC group. In fact, they're really most closely associated with the endothelium. And I can show you that in more details here. So when we do a correlation analysis, the primary cells hit the conventional DC subsets in the atlas, but their reprogrammed cells here all are very closely aligned to endothelium. They haven't quite reprogrammed the way they had hoped. So just to summarise that in that whirlwind tour of the Stemformatics Atlas, what I've shown you, I hope, is that, you know, these redundant platforms that we're moving away from, what they lack in cell resolution, we gain in molecular resolution, so they've got very, very strong sets of comprehensive coverage of the genome, and also experimental resolution. The metadata associated with them is really informative, and we can draw on that to understand better and annotate our single-cell data sets. It's important that we're combining data from many different sources when we're wanting to understand patterns of behaviour. I think that we've seen great examples of that, particularly in this session where we've been talking about integrating data from many, many different people. And when we're able to do that, then these anecdotal patterns in one data set start to become generalisable patterns across the atlas. That it's very useful, in particular, to be able to identify subsets which you are hoping to capture in single-cell data, but often get clustered together in an amorphous blob, hard to distinguish. So by using the reference atlas, we can tease out some of these subsets. And it's a useful approach, and this is what we use it for, to benchmark new models that we're deriving completely in the laboratory dish, and just to ask how well are we recapitulating human biology and the tissue-specific biology that we're interested in. So I'd like to finish my talk now just by acknowledging a couple of the key people in my group. So I spoke to you about the Stemformatics platform, and this is really the work of Jani Choi, who's the team leader of this group, ably enabled by Suzanne Butcher, who I'm showing you there with the kids. Suzanne has done all of the manual curation of experimental metadata in our atlas. I showed you the projection of single-cell data onto our atlas, and this is work that was done by Yidi Deng, a very talented PhD student who's jointly supervised by Kim Ann Lecau and Jani. And of course, I want to acknowledge Nadia Rajab, whose work I showed you on the human-induced pluripotent stem cell-derived macrophages. Thank you very much.
Thank you very much for the excellent talk. Any question from the floor? Yes, please.
Christine, great work. Thank you. very interesting about overlapping reprogrammed cells onto reference and finding that it's kind of falling short of achieving the desired cell type. Is your tool, or do you think there are any analytical tools to understand what factors are missing, and is there strategies to which we can add or modify additional factors so that you can actually reach the full conversion that you expect? And I think it would be really nice to have that for not just macrophages, but other cell reprogramming efforts.
Yeah, I completely agree with you, Jay. And so this is something that I didn't present today, but we are actively working on. So we can currently look at the experimental factors which accompany different reprogramming approaches to see whether there are additional growth factors in the media or timing effects which have an influence. We have run some network analyses to try and identify, for example, additional transcription factors. But to be honest, I think it is actually a combination of the transcription factors and the dish environment that's going to be really critical. And for a lot of these myeloid cells, I think it comes down to putting them in co-culture with stroma and T cells.
Great, thank you.
Christine, nice to over here. On your left.
Hello.
Okay, Jonathan from IMCB. So in terms of the difference between the differentiated dendritic cells to the primary tissues, how much is epigenetic memory come to play here?
It's a really great question because, of course, the induced pluripotent stem cell comes from a different type of cell, a fibroblast or a blood cell originally, and the reprogrammed dendritic cells that I showed you are all from fibroblasts. So it's pretty clear that they're remembering their tissue of origin because they're staying very close to those stromal cells. When we've compared iPS-derived macrophages from different sources, so for example, iPS cells that originally came from a dermal fibroblast or iPS cells that originally came from a lymphocyte, we don't see the epigenetic state of the pluripotent cells make a difference. So have the reprogrammed dendritic cells simply not overcome that epigenetic barrier? We don't have the data available to us to tell, but I suspect you're right. I suspect
because the transdifferentiations will not have undergone a lot of replication cycles.
That's right, yeah, exactly. It's a very short time window when you have a look at that experimental series, no more than about 10 days.
Hey, that was a really nice talk, and sort of echoing. It's a really great demonstration of how the easy-to-use visualization tools can drive a lot of insights. And so this idea that, thanks also for illustrating this sort of common pitfall for a lot of the classification techniques, that if you just don't have the right things in there, they're not going to you can get a spurious classification. So to maybe step back just a little bit from the questions you were getting just now, like on a more technical level, so you showed nicely that those sort of differentiated cells from that one paper were not near anything else. But then, if you go back and were you able to get their DC1 through DC6 populations and put them on, like, perhaps that those things already there was a big batch effect. Do you understand what I'm saying? Like, so those cells that you put in didn't look like anything that you had in your atlas, but maybe what they were classifying towards were also somewhat divergent from what was in your atlas.
Well, we did include in our projections the primary cells that they were comparing to in their TSNE, and their primary cells match the conventional DC subtypes exactly within our atlas. So I think what they were projecting towards was captured in the atlas, but unfortunately the route that they were trying to go there didn't quite make it.
Yeah. Okay, great, thanks. That's a really important result, so thank you for sharing.
My pleasure.
Well, thank you very much, Christine. The next speaker is Dr. Chung Xiaohon from Riken Center for Integrative Medical Science in Japan. He works on the origins and functions of non-coding transcription in human genome with a focus on long non-coding RNAs and enhancer RNAs. Please welcome Dr. Chung Xiaohon.
Thank you. Thank you for the introduction. And thank you for the organizers. Well done, Popan. Did a very good job. This is probably one of the best conferences I've been. Thank you. So today I'm going to talk about transcribed cis-regulatory elements, profiling those in the single cells, and how we can use those information to interpret disease genetics. So cis-regulatory elements generally refers to promoters, enhancers, and they regulate gene expression, apparently. And so these genetic variations lying in these cis-regulatory elements somehow contributes to disease dispositions. So in fact, if you look into the genetic associations studies, that the majority of the variants associated with the diseases is located somewhere in these cis-regulatory elements, mainly promoters and enhancers. And of course, a lot of these cis-regulatory elements, they are very cell type specific in terms of activity. So looking into those physiologically relevant cell types, single cells in particular, that would be essentially very relevant to understand disease disposition. So there are many ways to profile the activity of cis-regulatory elements, but today that we are looking into one particular method that measures the transcription output from these cis-regulatory elements. And by looking into the 5' end of the RNA produced from these elements, that you could precisely locate where the RNA came from, and hence also the location of these cis-regulatory elements. And it happens to be there are already available methods that you do not need any modification that are available on the market. So the 10x 5' end kit, that it happens to be the read 1, which contains the very first base pair of the RNA, which could be used to map the transcription start site, which in turns that to locate these cis-regulatory elements. So the problem is, here I'm showing you one examples, that the examples on the left-hand side. So the examples on the left-hand side is a promoter. What you're seeing here, right here, so is the single cell resolution, single base pair, single cell resolution signal that we get from the 10x kit. So and here is the pseudobulk putting everything together. As you can see, that precisely mapped to the lone transcription start site of a gene right here. On the right-hand side here, that you're seeing an example of an intergenic enhancers, which is showing these distinct bidirectional transcription patterns. And again, that you're seeing here single cell resolution, single base pair resolution of the transcription pattern. And here that you're seeing the pseudobulk is bidirectional, but at single cell actually they are unidirectional. So the problem is that how could we extract those information in a reliable way? So it turns out that the 5' signal that we get from the unprocessed 10x reactions, that you get a lot of noise, that when you do a plain clustering, that most of the clusters is actually not really representing the genuine transcription start site. So, and we wrote a little software called Escafe, which allows you to feed in directly the 10x BAM files, identify the TSSs, and distinguishes noise from signal based on a multiple logistic regression classifier that we built using epigenome information. So if you happen to have some 5' end data, that you could try this software, that you could extract these regulatory elements from your 10x data without additional cost. So you don't have to buy a taxi kit for that. Okay, so. Here, I'm showing you some examples, locus that you could recover from the 10x reactions. So right here, there's a gene transcribing from right-hand side to the left, and here is the promoter of the gene, and these are the elements. somewhere located close to the gene. Some of them are at the intron, some of them are at the intergenic region, downstream of the genes. And here that you're seeing, this is the ataxic signal. So these are two reactions that done in parallel with the RNA and the ataxic kit. Both are single cell on the same cell line, so that you're seeing the signals are pretty consistent with each other. And for the ataxic CREs, we call them accessible CREs. You could also detect super enhancers. So here is just one locus located at the introns of a gene. That here you're seeing these signals. This is the signal that we get from the Tanix, and some of the other tracks showing here is consistent with the signal we get from the Tanix. So that's the Tanix 5' kit could also detect super enhancers. So here in the following, I'm trying to compare that how does this two technology, ataxic and the 5' kit, behaves in terms of measuring the activity of these regulatory elements in single cells. So we are using a system with PBMC under stimulation. So the first thing we look into is how does this cell type specific CRE looks like between the two sets of data. That without surprises, the activity of this cell type specific CRE is very consistent between the ATAC and the 5' kit. So, but when we look into more details into some more subtle changes at the cis regulatory elements upon stimulation, that we are seeing some extra resolutions. Say, for example, here there's this one gene that has two promoters, and we are seeing a usage shift in the RNA expression, but not in the ataxic data. And those shifts are very cell type specific also, so that we are concluding, and perhaps that the RNA is much more quantitative when you are trying to look into very subtle changes between cell state. So, and the other thing that we could do is actually to link this regulatory element, which is far from the genes, and to the gene promoters by coactivity. So this is how we're going to interpret the GWAS signal that is far from the gene. For example, right here, I'm showing you one locus, and all these blue bars right here are the disease-associated SNPs, which is located essentially in the region void of genes. But there is a super enhancer located right here, and this super enhancer is co-expressing with the promoter of the gene right here. So the correlation looks very genuine right here. So, and we're comparing how much of these connections that linked by coactivity that could be validated by high-C data. And we are seeing a much more likelihood of being validated in the RNA data than the ataxic data. So right here on the right-hand side. And it's probably also due to the more quantitative nature of the RNA data when you compare with the RNA-seq data, with the ataxic data. So the other thing we look into is how much of the disease heritability is enriched in these regulatory regions identified by the ATAC and the RNA-seq. So the first thing we see is there's much more enrichment in the enhancer regions, which is in the lower row right here. And those enrichment seems to be a little bit higher in the RNA when you compare with the ATAC-seq data. So we try to break it down into something further. We look into the ATAC-seq peak that has the RNA-seq signal versus the one that does not have the RNA-seq signal, which is actually the majority. So, and those those ATAC-seq peak that does not have the RNA signal is actually virtually not enriched with any disease heritability. So our conclusion is maybe the RNA, the transcribing as its regulatory element are more relevant in terms of a disease predisposition. So we take this thing we learned from the TCRE, and we try to profile these TCREs in a wider scale. So actually in collaboration with many different universities in Japan, that we collected 23 tissues, and after filtering that we obtained about 500,000 cells. And by looking into the cells that we defined about 200,000 cis regulatory elements, and most of them are enhancers. So this is what the data looks like. 200 broad cell types and 128 the narrow cell types, which has a pretty good coverage of the major cell types that you could find in human body. The question is, how could we leverage this TCRE atlas to understand the disease heritability? So we came up with a method, a score called ice cream score, individual cell enrichment of cis regulatory element activity module score. So what we try to do is we try to come up with one single score that you could project the disease heritability into every single cell in the data set. So essentially, there's 4 steps in the procedure. I will briefly go through that quickly. So the first is we have to define the activity modules, CRE activity modules, pretty much like what Zhong'an did with the NMF modules. So the first thing we do is to collapse the data a little bit so that from 500,000 cells to about 3,000 metacells to make it a little bit more affordable computationally. So, and we identify the CRE activity modules using this metacell data set, and we define about 230 CRE modules. Essentially, those modules are reflecting the cell identity and the cell activity. So, and then we look into the disease heritability enrichment in each of these modules, and you see a very specific pattern in different modules across these 30 diseases we've tested. And the next thing we try to do is we try to combine this per module disease heritability enrichment and project this per module activity back to the single cells. So in terms, then you get something like a weighted heritability enrichment module activity on a per cell basis. And lastly, we do a permutation on the CRE labels to get the score versus the low distribution. So, and this is what it looks like. Just show you a couple of examples right here. So this is an example of a bone density GWAS result projected back to the atlas that we made using the ice cream score as I described. And you can see that kind of makes sense that this bone density heritability is very enriched in a subset of the chondrocyte. And of course, we look at other diseases like neurological diseases, a psychiatric disease. For example, right here, bipolar disorder, that you're seeing there's an enrichment of the heritability in a subset of oligodendrocytes, progenitors here, as well as a subset of neurons right there. So one more example. So this one is a little bit more systematic, more diverse, involving multiple cell types. Psoriasis, autoimmune diseases on skin. So epithelial cells, you can see here, endothelial cells, immune cells. But in particular, let's zoom into one of the example. So still psoriasis right here, so that you can see there's only a very specific, a small subpopulation of endothelial cells, which is the vein-like endothelial cells is enriched in heritability. So if you look into different types of different diseases involved potentially with the endothelial cells, you see a very different distribution, and all of them is showing in a only very restricted subtype of endothelial cells. So, and what we're gonna do with these things, right? So eventually, that if you have a ice cream score, which is on a per trait basis, that you could correlate this score with different features that you collect on a single cell basis, and say, for example, here that I'm correlating the super enhancer expression with the ice cream score of this varicose vein, and you see there is a pretty good correlation between these two. And this super enhancer itself is also overlapping with the disease-associated SNP with varicose vein. So these are the examples that I'm showing to you, and how we could use the TCRE atlas to interpret the disease heritability. So all this work, that is a collaboration with different universities in Japan that's generously donating all these samples and collaborating with us. And most of the work that you're seeing is done by Jonathan Modi, a postdoc of our lab. And the experimental work was mainly done by the researchers in Jason's lab. So, and we are hiring, if you're interested in sushi, hot springs, and come and chat with me. Thank you.
Thank you very much for your wonderful talk. We have time for one question from the floor, please.
Great talk, Cece. So can I ask you to elaborate on something you mentioned in passing right at the beginning of your talk, and that was that a lot of elements which look to be bidirectionally expressed in bulk are only expressed unidirectionally on the single cell. Can I ask, how cohesive is that? So do you see preference for one direction coming out of, for example, the enhancers? And can you comment on whether you see exclusivity of events which are sitting very closely, like head-to-head type coding, non-coding transcription?
Yeah. So these questions are very meaningful, but it's very difficult to be investigated, mainly because that you get very limited number of read on a per locus basis in a single cell. That the one example that I'm showing is one of the very few examples that you could have this many signals. That we apply a filter of more than 20 read per cell in the locus to get these signals. So to answer that question, we have only a very few number of locus that actually have enough signal to investigate. So my answer to your question is we do not have any statistical power. We only look at specific samples, 10, 20 of them, but we don't find anything that is meaningfully correlated with where it's located, upstream or downstream of the gene, how wide is the enhancers. There's nothing that we found is correlated. But one thing that worth considering is anything that we are seeing here that's supposed to be, very bursty expression with the enhancers. And if you consider there's two alleles, that it doesn't make sense that only one allele is expressing, and only one direction is expressing, even you have two alleles, right? So, and what we think it could potentially be only, because of this bursting nature, that what we're capturing is only the transcriptional output from one single allele, that what we think. Yeah, but other than that, there's no further information that we could look into because of the amount of the signal we have.
More data.
Yeah.
Hi, Angela Wu from HKUST. Very nice talk. I think you know Danny from our department pretty well. So yeah, really interesting data, especially at the end when you presented the kind of how you identify the subsets of cells which are related to the specific disease. One thing I had in mind is that currently it's very difficult to isolate the subpopulations or the so-called narrow subpopulations of cells. Do you have any ideas about how you might follow up on this data with functional studies? That's something of interest to us. So just wanted to see your thoughts.
Yeah. Good question. So it's a very good question. It's also difficult to answer experimentally. So it depends on when you say isolate particular cell type, it depends on you say isolate like physically from the tissue, or you want to isolate, the data either from the library itself or from the, or from the BAM file, right? So isolating particular cell population from the physiological tissue, I couldn't give you, any answer on this. Me myself is more computational. Okay, so, but I discussed with Alistair Forrest the other day, actually yesterday, that he was talking about that if you really are really interested in one single cell type, and you want a much deeper, transcriptome of that particular cell type from your data, that potentially one way of doing this is you could actually capture the cell type by capturing the cell barcode. Okay, so say for example, if you want to look into long read splicing isoforms of that particular cell, that if you have the short read data already available, one potential way would be capturing the cell barcode which you already sequenced from your short read data to get a deeper sequencing or with a longer read on those cell populations. I'm not sure that answers your question, but this is on the molecular level, not the physiological level.
Yeah. And also just to kind of follow up on Christine's question about the allele-specific expression, I think Richard Sandberg recently did some work on that, which you might be interested in. So anyway, look forward to chatting with you more later.
Thank you.
Thank you very much, Dr. Chung Xiaohong. So that comes to the closure of our morning session.